Okay, good morning, everyone. Since our projector still isn't working, I'm going to go the route of presenting you this information in this short little video. So let's get going. Okay, so section 8.4 is all about transcription. Now, what is transcription? Okay, transcription is a part of a process that we're going to look at of how we go from a DNA information, an instruction to the actual product, right? It's like going from the instructions of how to build a bookcase to the actual bookcase. So that's what we're going to do first, okay? So let's go to the first slide. Okay, so shortly after his discovery of the structure of DNA, Dr. Francis Crick came up with something called the central dogma. Now, the central dogma tells us that information flows in one direction, and as we can see here, that information is flowing from DNA to another nucleic acid called RNA, very similar, and we'll talk about the differences in the next slide, or in the next couple slides, and then finally to a protein. Now remember, a protein is the molecule that's going to do the actual work. Okay, so let's now we have steps for this as we move from within the central dogma. We have steps as we move from DNA to RNA to protein. Okay, so as we move from DNA to RNA, we're going to go through the process called transcription. Now, what's transcribing mean? Think about what transcribing means. Transcribing means you're going to rewrite something and maybe change a little bit, right? So like when you are taking these notes, you're actually transcribing them, right? It's not going to look exactly like it is on the screen here, right? It's not going to look exactly the same, but it's going to look similar. Right? As you take notes, you're making them close to the same thing, but not exactly the same thing. And that's what's going to happen as we transfer from DNA to RNA. We're going to see a slight change in the wording or in the code for how we write things. Oh, people are going to start walking the room here pretty soon. I got to hurry up. So what makes DNA and RNA different? Let's look at that. Okay, so first of all, first thing is part of the backbone. Okay, in RNA, okay, you'll notice that the backbone is different. Okay, if you remember, it was pentagonally shaped, five-sided shaped in DNA, but in this one, we see that it's six-sided. We'll also remember that there was an oxygen right about here in DNA, and then there was a carbon up here. Right now we see these two methyl groups here. Okay, so and that's why we say that DNA is ribose nucleic acid, right? Or ribonucleic acid versus deoxyribonucleic acid like DNA. The other thing is RNA doesn't use thymine, T. It uses uracil, okay? And then the last difference is right here. RNA is single-stranded. If you remember, DNA is double-stranded, okay? And that the reason for that will be more important, and you'll see why that's so important when we go to protein synthesis or that translation. Okay. In transcription, what's going to happen is we're going to read and then transcribe a strand of one of the strands of DNA and make a single strand of RNA. Okay, so how does that process go? Let's go into detail of how transcription comes along. Okay, so first of all, transcription still has some proteins that are similar, but some proteins that are enzymes that are different. So first of all, helicase still has to come in here and unzip the DNA. You still have primase is still going to come down, but instead, in replication where we had DNA polymerase, we're going to have RNA polymerase. RNA polymerase is going to be that one that reads the DNA code and then attaches the appropriate RNA nucleotides. Okay, and as I said, it recognizes the gene that it needs and unwinds it. Okay, with helicase. Here comes kids. It's going to get loud in here. Sorry, but I'm trying to go fast. Okay, so as RNA reads the DNA sequence, it's going to pair up the correct nucleotide. C's and G's always go together. Anytime it sees a T, it's going to put an A, but when it sees an A on the DNA strand, it's going to put a U on the RNA strand. So do remember that. Now, I have here an animation. We'll see if it works. I don't know if it's going to work. You going to play for me? Please play. Play. I don't know what we're going to do. Let me try something. just do this. Try some. I don't know if it's going to work. Oh, come on. Come on. I know I'm putting a heck of a load on my computer right now. It's not going to work. Crap. Alright, if this animation doesn't work, no biggie. I will just... That's not going to work. I'll just send this to you guys separately, okay? Okay. Alright. So, the RNA, once it reaches the end of the gene, and there's actually a specific code on the end of a gene that tells the RNA polymerase to let go. And when it lets go, it breaks apart. Now remember, I didn't tell you this part, but RNA is actually a bunch of different enzymes that come together. We call it an enzyme complex, often in biology, but then it falls off. Okay. Now, in this process, we're going to make one of 3 different types of RNA. We're going to make messenger RNA. Now, this stuff right here, mRNA, is going to be really important because this is actually what we're going to make the proteins out of. Okay. There are codes for making ribosomal RNA within your DNA sequence, and this is what's going to make ribosomes, the proteins that make up ribosomes. And then lastly, there's transfer RNA, and this is all. A messenger RNA and transfer RNA kind of go hand in hand. Transfer RNA, as we'll see in translation, is the one that's going to attach to amino acids and then bring them into the ribosome, it's going to bring in the ribosome and eventually build the amino acid. More on that later. Okay. Last slide. Thank you. Yeah, last slide. So the transcription process is very similar to replication. As I told you before, helicase is involved, primase is involved. Transcription and replication both involve complex enzymes, like I just said. You know, RNA polymerase, DNA polymerase, very similar. And in either case, we're going to get a copy of a gene sequence or of a nucleic acid sequence, to be more exact. Okay. Now, what's the difference, right? Big difference is, we have two major big differences, right? First of all, in RNA, we're going to use uracil instead of thymine. And then the second difference is we're going to only make one single strand, whereas in replication, we're going to make 2 double strands. So, a little bit different. Yeah, sorry about the video didn't work. That's all I got for today. I will send all this out, good Lord willing, to you and get it posted on Google Classroom. If you have any questions, let me know.